welcome to the world of biology through Jaguar classes from Jagran Public School, Noida. In today's this class or this lecture of class 12th, I will continue with the unit 7 genetics and evolution, in which we have uh, taken the chapter 5 principles of inheritance and variation, and in this part 8 of this chapter I will cover the topics autosomes and sex chromosomes concept, linkage and its types and significance of linkage, crossing over and its mechanism and types, and chromosomal gene mapping and its significance in understanding the concept of principles of inheritance and variations. After today's class, the expected learning outcomes, knowledge based category, students will be able to know and recall the concept of autosomes and allosomes, describe and compare the phenomenon of linkage and recombination, and in skill based competencies you will be able to compare and describe complete and incomplete linkage. You will be able to critically analyze the phenomenon of linkage and recombination with the deviations occurred in Morgan's experiment in Drosophila melanogaster, and side by side these knowledge based and skill based learning outcomes, the competencies that you can inculcate, collaborative learning, critical thinking, observation and analytical skills to solve various types of HOTS and case study based exercises related with the linkage and recombination concept. Now let's start with our today's class. And in today's this class of principles of inheritance and variation, we will focus on the concept linkage and recombination, which is also one of the important concept or phenomenon related with the principle of inheritance. In this class, we will cover the topics autosomes and sex chromosomes, linkage, its types and significance, crossing over and its mechanism and types, which is the uh, basis of linkage and recombination, chromosomal gene mapping and its significance. Now let's start with this slide. With the help of this slide, you will be able to understand and recall the concept of types of chromosomes. As in your earlier class, in class 10th, you have learned about the autosomes and allosomes. Autosomes are the somatic chromosomes which determine the body characters of an organism. And as you know, in humans, there are 22 pairs of autosomes which are responsible for our vegetative features, or we can say that somatic features, which are non-reproductive features. And allosomes or sex chromosomes are the chromosomes which determine the sex of an organism. However, they also have genes that control various body characters like sex-linked characters. And humans have a pair of sex chromosomes, XX in females and XY in males. And here you can see one human karyotype, and karyotype is a pictorial representation of all the chromosomes mapped in different numbers from 1 to 23 numbers of pair of chromosomes that you can see in our karyotype, where we can see that how these chromosomes are located and these chromosomes are present in our cells. Now here in this slide you will be able to understand the sex-linked inheritance. In the concept of inheritance, when we are talking about sex-linked inheritance, as its name suggests, the body characters which are determined by the genes on the sex chromosomes are called sex-linked characters, and their inheritance or transfer of these characters, sex-linked characters, from one generation to another generation or from parents to their uh, children, known as sex-linked inheritance. Thomas Morgan was the biologist who has first reported the inheritance of sex-linked characters and their association with sex chromosomes in Drosophila melanogaster, which is also known as fruit fly. Here you can see that X and Y chromosomes, which determine the gender in case of Drosophila, and later on it has been studied that in humans also the same phenomenon is used for the sex determination on the basis of genetics. Gene for color blindness is also present on this chromosome, uh, which determine the body character like color blindness, and that's why color blindness is also known as X-linked or sex-linked inheritance. Now we will understand about the linkage. This is a phenomenon where we can see that when two or more genes on the same chromosome tend to be inherited together, these genes are called linked genes, and the phenomenon is called linkage. When these genes are located on the sex chromosome, then they are called as sex-linked genes. As in this uh, chart, you can see that how the linked genes are moving together from one chromosome to another or from one generation to another generation, because they are located at one place. Now, types of linkage. There are two types of linkage. One is considered as complete linkage and another is considered as incomplete linkage. Have a look here in this chart to compare about these two types of linkage. In case of complete linkage, that results from when genes are present very close to each other, while incomplete linkage takes place when genes are separated during crossing over in gamete formation. Complete linkage results in parental combinations, and incomplete linkage results in new combination in the progenies. Example of complete linkage you can take as grey body color and long wings in male Drosophila. While the examples of incomplete linkage we can see in sweet pea variety with blue flowers and long pollen. The significance or utility of this linkage is it reduces the variation, thereby keeping parental combination together, and the same combination of characters also appears in the offsprings. It also prevents the introduction of new varieties in plant breeding program. Now here let's learn more about the complete linkage with the help of this chart. In complete linkage, genes are tightly linked, which resulting in very low percentage of recombination. Like in this case, parental generation is showing whatever type of genotype here, and in F1 generation, when they are moving from this generation to this generation, and in F1 individuals, when recessive parent get uh, crossed, then F2 generation back cross phenotypes, we can see that 50% type of uh, purple and blue color we can see here with the help of these genotype transfers. In case of incomplete linkage, the same feature we can see here. In incomplete linkage, genes are loosely linked. For example, here the two genes color and seed shape, you can or pollen shape, we can see here are the two characters which are showing incomplete linkage, and here we can see that genes are loosely linked on chromosomes and they show higher percentage of recombination. As in case of this example, we can see that in F1 generation, blue flower long pollen plant when crossbreeded or test crossed with the pink flower round pollen, Then there are four types of combinations in F2 generation. In progenies, we can see blue flower long pollen 43.7%, blue flower round pollen we can see 6.3%, and pink flower long pollen 43.7%, and pink flower round pollen 6.3%. These were the results obtained by uh, different biologists time to time when they have performed the experiments related with the linkage. Now, the basic phenomenon or basic concept which is related with the linkage or recombination that we can see here, crossing over or recombination, due to which new recombinants will form in the progenies. Recombination is an event that occurs during meiosis where homologous chromosomes exchange genetic material. As you can see here, these are the homologous chromosomes, and these sister chromatid when get crossed over, they will from here when these homologous chromosomes crossed here, and then this exchange of chromatin material get changed in these two sister chromatids, and as a result the gametes will be of four type, and out of them 50% are parental type and 50% are recombinant type with new genetic material. There are few recombination if the linkage between genes is strong. Means if they are closely associated, two genes are closely associated, then recombination will be few, and recombination will be of high degree or a high percentage if the linkage is weak. Means two genes are situated apart from each other or at longer distance. Now try to understand and once again recall the concept of mechanism of crossing over at the time of meiosis first stage. Here synapses that we can see that here the two homologous chromosomes paired lengthwise resulting in bivalence, and duplication of chromosomes takes place when each homologous chromosome splits to form two sister chromatids. In this chart you can see that attached at their centromere. These are the two sister chromatids which are attached at this centromere, and chasmata formation will take place between these two sister chromatids of two homologous chromosomes. The point of and that chasmata is actually the point of contact where the non-sister chromatids of homologous chromosomes exchange their genetic material is called chasma, and it takes place in Pachitin stage of the meiosis first in case of meiosis cell division. Now let's learn about types of crossing over. The crossing over can be of these three types. Single crossover. Here you can see that single chasma is formed resulting in single crossover gametes, and this type of crossing over frequently occurs in the organisms. Double crossover, where we can see here two chasmata are formed resulting in double crossover gametes. And multiple crossover, here you can see that 1, 2, 3, 4, 5, 6 more than two chasmata are formed, and that is rarely occurring stage or rarely occurring type of crossing over. Now try to understand the significance of crossing over for the organism. By this process, the redistribution of different traits to different individuals within a population takes place, and which brings about variability in the genotypes of different sexually reproducing organisms, and useful for plant and animal breeders to introduce new desired characters. Now, one more concept related with the study of linkage and recombination by Morgan and other scientists, which were the co-worker of the Morgan, that is known as chromosomal mapping. The position of the genes on a chromosome is always fixed, and chromosomal mapping is a technique by which we can show the sequence of genes on a chromosome that you can see here. Different band-like structures are showing the sequential manner of different types of genes located on these particular chromosomes. And they are also showing the distance between the genes we can easily see from here. And scientists use a map unit to describe the distance between linked genes, and this unit is called Morgan unit because it is first time uh, introduced by the Morgan. And in that chromosomal mapping, when we will understand about the significance of chromosomal mapping, the four areas where we can use or utilize or apply this chromosomal mapping. First of all, it helps in predicting the result of untried crosses. Second field where we have applied the chromosomal mapping technique, it helps in finding the exact location of a gene on a chromosome during genetic discovery. Well, it also validates genes located at specific loci on chromosomes, and the maps can show if any genes are missing from a chromosome. Now have a quick recapitulation of today's topic that we have covered in our today's class. One point that we have discussed, autosomes are the somatic chromosomes which control the expressions of the body characters other than the reproductive character. Sex chromosomes are the chromosomes which determine the gender of an organism. However, they also have genes that control various other body characters also, which are known as sex-linked characters. Linkage, the inheritance of genes of the same chromosome together, and the capacity of these genes to retain their parental combination in subsequent generation is called linkage. Recombination is an event which takes place between homologous chromosomes, resulting in an exchange of genetic material. And chromosomal mapping, that is a technique which shows the sequence of the genes on a chromosome and the distance between the genes. Scientists measure the distance using a unit called Morgan. I think with that we have covered up our today's class topic, but I am showing here one presentation related with the today's topic, linkage and crossing over. Before going to show that presentation, here is one discussion question: chromosomal mapping. How can chromosomal mapping be used to improve human health? You have to write about it, and you can use these clues uh, which can you write in your answer. Inherited disease causing genes can be identified and mapped by following the pattern of inheritance of a DNA marker, and a DNA marker which is linked to the that particular disease causing gene would be present in affected individuals but absent in unaffected ones. And in this way chromosomal mapping help us to understand the various genetic factors that we can study to study any uh, hereditary disease. Now, I'm starting this presentation with the help of this media presentation. You will be able to clearly understand the concept of linkage and recombination.
Linkage and recombination. In the early 20th century, Thomas Hunt Morgan made an insightful choice of using Drosophila melanogaster, the fruit fly, to further understand Mendel's findings. The fruit fly usually has red eyes, but in 1910, Morgan found a male fly with white eyes. He crossed this mutant male with a red-eyed female. All the offspring in the F1 generation were red-eyed. The F1 generation males and females were crossed. The white-eyed trait appeared in the 3:1 ratio, same as the Mendelian ratio. However, only the males were white-eyed. This suggested that the white-eyed trait was present on the X chromosome. Probably the red-eyed females were pure for the trait. Since all the flies were red-eyed in the F1 generation and the ratio of red-eyed trait to white-eyed trait was 3:1 in the F2 generation, Morgan concluded that the red-eyed trait was dominant over white-eye. Morgan further crossed the F2 males and females. Eye color in males depends on which X chromosome they inherit. A white-eyed female has recessive traits on both X chromosomes. Since Morgan found eye color trait to be linked to the X chromosome, he called this inheritance pattern as sex-linked inheritance. Morgan conducted a dihybrid cross similar to Mendel. He crossed the wild-type red-eyed, large-winged males with mutant females, having white eyes and miniature wings. In the F1 generation, only the parental combinations appeared. But in the opposite sexes, the males were white-eyed, miniature-winged, and the females were red-eyed, large-winged, in contrast to the parents. Then Morgan hybridized the male and female flies from the F1 generation. According to Mendel's law of independent assortment, the parental traits should independently assort to the gametes. Therefore, it is expected that 50% of the progeny carry the parental traits and 50% progeny show non-parental traits. However, the actual results deviated quite a lot from the expected. About 63% of the progeny carried the parental traits, while only 37% showed non-parental traits. What could be the reason for such deviated results? Let's try to understand.
Now, what is the reason behind this
To unravel
mystery?
the mystery of the deviated results of Morgan's cross, let's take a look at the arrangement of genes on the X chromosome of the Drosophila. There may be a few hundreds or even thousands of genes located linearly on a chromosome. According to Mendel's third law, the genes responsible for two different traits assort independently. But actually, this can happen only if the genes are on different chromosomes or far apart from each other if they are on the same chromosome. The genes present far apart on the same chromosome get recombined by crossing over during meiosis. This was what happened in Mendel's experiments, helping him to arrive at a consistent set of results. But in Morgan's experiment, the genes for the Drosophila's eye color and wing size were located not very far apart on the X chromosome. Now, crossing over can take place anywhere on the chromosome, and it doesn't necessarily lead to recombination. Morgan termed this condition as linkage and the genes as linked genes. However, when crossing over takes place near the alleles for eye color and wing size, the alleles segregate to different gametes. This results in non-parental combinations, which Morgan called recombinant types. Since the frequency of crossing over of linked genes is less, parental types appear more often as compared to the recombinant types in the F2 generation.
Now, you can see this example for which the hint is given here to solve the mystery. Of the 7 traits in garden pea that Mendel studied, 2 of them are in chromosome 1 and 3 of them are in chromosome 4. Yet he got consistent results of 50% parental combinations and 50% recombinations, unlike Morgan, who got varied results for linked genes. What could have what could have happened in Mendel's case? You can answer this question with the help of this clue or hint. In Mendel's case, actually, the genes of those traits are far off from each other, and every time they would have crossed over and assorted independently, which were showing that result of 50% parental combination and 50% recombinations.
Morgan continued to experiment with Drosophila by conducting yet another dihybrid cross. White-eyed, yellow-bodied females with red-eyed, brown-bodied males. Brown body and red eyes are dominant traits. Yellow body and white eyes are recessive traits. In the F1 generation, the frequency of parental traits was 50%. In the F2 generation, again, the parental combinations were more frequent than the recombinant traits. However, the deviation was much more drastic. The parental traits were intact in almost 99% of the progeny, and only a negligible 1% showed recombination in traits. This happened because the genes for eye and body color are located quite close to each other. Hence, even if chromatids cross over, the gametes get the parental combinations only. Thus, Morgan concluded that the frequency of recombination depends on the relative location of genes on a chromosome. The closer the genes, the lower the recombination, and vice versa. From the 2 crosses conducted by Morgan, we can understand that the genes for eye color and body color are more closely linked to each other as compared to the degree of linkage between eye color and wing size. In humans, genes associated with cystic fibrosis and color vision deficiency are linked. Alfred Stutwint, one of Morgan's students, considered the frequency of recombination between gene pairs as a measure of the distance between genes and mapped their position on the chromosome. Thus, concluding from the crosses we saw earlier, since genes for eye color and body color segregate only on 1% of the occasions, Stutwint considered them to be one map unit apart. Similarly, we can say that the genes for eye color and wing size are 37 map units apart.
And in this way, the Morgan and his student given the concept of gene mapping. In a hypothetical cross made between two insects, the recombination frequencies between genes A and B is found to be 27%, between BC it is 63%, and in AC it is 36%. Can you map their loci on a hypothetical chromosome? Yes. Whatever will be the percentage, that will be the map distance between these two genes. For example, if AB is found to be 27%, means the distance between B and A is 27 map unit, and in B and C we can consider it as 63 map unit distance will be there, and in between A and C, due to 36% results here, we can consider or conclude that the distance between A and C is 36 map unit.
Let's summarize what we have learned so far. Click the tabs to learn more.
These are the three areas on which we have focused in this presentation: Morgan's experiment, crossed eye color and wing size, that you can see, in which Morgan experimental cross eye color and wing size, that result has been seen, and in F2 generation observed frequency of parental phenotypes and recombinant phenotypes is 63% parental phenotype and 37% recombinant types. And in Morgan's experiment or experimental cross of body color and eye color, the results are shown in this chart. Here, the parental phenotypes observed 99% and 1% recombinant phenotypes were observed in that case. And in third case, we can understand about the gene mapping. With the help of this chart, we have understood about the gene mapping and relationship between the percentage of parental type and recombinant type to find out the distance in the form of gene map unit. We can see. Now, I hope that that concept linkage and recombination is clear to all of you. With that, I am finishing our today's class.